Hello friends, in this video we'll study about synaptic transmission, summation and termination. Synapses in vertebrates, especially humans, are chemical in nature, while those in lower animals they are electric in nature. Presynaptic neurons synthesize neurotransmitters at synaptic terminal, which is packed in synaptic vesicles. In this diagram, we see these are the synaptic vesicles, and this red colored neurotransmitter is lying inside the synaptic vesicles. Okay. On arrival of action potential, voltage gated channels open and calcium diffuse in, as a result of which there is high concentration of calcium ions in the synaptic terminals. We see here calcium is moving in. Okay. Vesicles fuse with terminal membrane, as a result of which neurotransmitter is released. Let us have a look at the diagram again. These vesicles, they are fusing with the membrane, and these neurotransmitters, they are released in the synaptic cleft. Okay. This neurotransmitter diffuses across the synaptic cleft and reaches the postsynaptic membrane, and the neurotransmitter binds to specific receptor in the membrane and activates it. Now, at some synapses, the receptor protein binds to neurotransmitter. It is a ligand-gated ion channel. They are known as inotropic receptors, and they are permeable to potassium and sodium. In this diagram, we see these inotropic channels, ligand-gated ion channels. This is the neurotransmitter over here, and sodium and potassium, they are moving in and out, so they are permeable to sodium and potassium. The channels are opened and ions diffuse across the postsynaptic membrane. As a result, a postsynaptic potential is generated. This is a graded potential in the postsynaptic cell. Such a potential is known as EPSP, or excitatory postsynaptic potential. Some ligand-gated channels are permeable to potassium or chloride only. Opening of such channels causes hyperpolarization, and this is known as IPSP, or inhibitory postsynaptic potential. Now we study about summation. Input from a single synapse is not sufficient to start response in a postsynaptic neuron because it becomes weak while spreading. Sometimes postsynaptic potentials combine to produce a larger potential. This is called summation, and this summation can be temporal or spatial. What is temporal summation? If second EPSP arises before the postsynaptic membrane potential returns to its resting value, both EPSP add together. Let us have a look at the graph. This is temporal summation. This is E1, this is E2, and this is the action potential. What is spatial summation? Sometimes multiple synapses are involved on the postsynaptic neuron. If such synapses are active at the same time, the resulting EPSP adds together. This is known as spatial summation. This graph shows spatial summation. E1, E2, and this is the action potential. Two or more IPSP, or inhibitory postsynaptic potentials, can also cause summation. They have a larger hyperpolarizing effect than a single IPSP. And all summations finally get affected at the axon hillock. This graph shows a diagram in which we have sub-threshold summation. They're not enough to cause action potential E1 and E2. This graph is showing special summation of EPSP and IPSP. This is excitatory and this is inhibitory, and they both add together E, E1 and I. So it is a special summation of EPSP and IPSP. Now, termination. The neurotransmitter molecules need clearing from the synaptic cleft to terminate the triggered response. Two methods are mainly involved in this process. There can be enzymatic hydrolysis and recapturing into the presynaptic neurons. After reuptake, neurotransmitters are repackaged in synaptic vesicles or transferred to glia for metabolism or recycling to neurons. If this process of clearing the neurotransmitter is blocked, consequences can be severe. For example, the nerve gas serine, it can cause death as it inhibits the enzyme that breaks down the controlling skeletal, the neurotransmitters which control the skeletal muscles. This is how termination takes place. Now we study about the neurotransmitters. The neurotransmitters are acetylcholine, then we have amino acids, then biogenic amines and neuropeptides. So first of all, we study about the acetylcholine. Acetylcholine functions which include muscle function and learning. They have acetylcholine as neurotransmitter. Acetylcholine receptors are of two types. They can be ligand gated or they can be G protein coupled. Ligand gated ion channels, they are found at neuromuscular junctions. And G protein coupled receptors, where they are found at the CNS and heart muscles. Okay. In heart muscles, acetylcholine released by neurons activates a signal transduction pathway. The G proteins in the pathway inhibit adenylyl cyclase and open potassium channels in the muscle cell membrane. These both effects reduce the rate at which heart pumps. Thus, the effect of acetylcholine in heart muscles is inhibitory and not excitatory. Okay. Nicotine, it mimics and alters acetylcholine functions. It acts as a stimulant by binding to inotropic acetylcholine receptors in the CNS. Botulinum toxin, they inhibit presynaptic release of acetylcholine, as a result of which we have food poisoning known as botulism. Botox is the brand in which injections of botulinum toxin are used to minimize wrinkles around eyes. And this is the structure of acetylcholine, as you can very well see over here. Now we study about amino acids. Amino acids are glutamate, glycine, and GABA. Glutamine is an amino acid which acts as neurotransmitter. It is the main neurotransmitter in invertebrates instead of acetylcholine, and it is main neurotransmitter in the CNS invertebrates and plays a key role in long-term memory. Glycine, it is an inhibitory neurotransmitter in the CNS and at inhibitory synapses in parts of CNS that lie outside brain. Then GABA, gamma aminobutyric acid. It is an amino acid and it is a neurotransmitter within the brain at most inhibitory synapses. When GABA binds receptors in postsynaptic cells, membrane permeability to chloride ions increases. This causes inhibitory postsynaptic potential. The example, diazepam. This drug binds to a site on GABA receptors, and the response, as a result of this, the response to GABA will increase. And this is how diazepam helps to reduce anxiety. This is the clinical use of diazepam. Now, biogenic amines. They are synthesized from amino acids, and we have three biogenic amines: norepinephrine, dopamine, and serotonin. So, norepinephrine, it is made from tyrosine, which is and this is an excitatory neurotransmitter. Norepinephrine is an excitatory neurotransmitter in the autonomic nervous system. ANS is a branch of PNS, peripheral nervous system. Norepinephrine also works as a hormone outside the nervous system and mimics the functions of chemically similar biogenic amine, epinephrine. Okay. Now we study about dopamine. Dopamine is made from tyrosine, and serotonin, this is made from tryptophan. Okay. Both dopamine and serotonin affect sleep, mood, attention, and learning. LSD and mescaline are psychoactive drugs which bind to brain receptors and produce hallucinatory effects. Biogenic amines have an important role in the treatment of many nervous system disorders. Example, Parkinson's disease is due to lack of dopamine in brain. Now we have neuropeptides. Neuropeptides act via G protein coupled receptors, and neuropeptides, they are actually short chain amino acids. The examples of neuropeptides are substance P. Substance P is an excitatory neurotransmitter for pain perception. Pain perception. Second is endorphins. Endorphins are natural analgesics, and they are released during physical and emotional stress. Okay, so this was all about synaptic transmission, summation, and termination. Thank you for watching the video. Thank you, friends.